Okay, so we have reached the final talk of day one, and it is my extreme pleasure to introduce you to Dr. Richard Bedlack, who is the neurologist and director of the Duke ALS Clinic, as well as an associate professor of neurology at Duke University, and the chief of neurology at Durham Veteran Affairs Medical Center. He is the head of ALS Untangled, which has published 41 open access reviews now. Many of them are the most viewed on the ALS FTD journal, so very, very highly accessed by the public, which is great. Also leads what we'll hear about today with ALS reversals. He has been named Best Neurology Teacher at Duke. He's been named one of America's Best Doctors, the American Association of Neurology Patient Advocate of the Year in the past. And it's a shame to put him on a virtual forum because Dr. Bedlack usually comes looking very fashionable, so I apologize to him for putting him on a virtual forum. But we're really looking forward to his talk, so without further ado, Dr. Bedlack, the floor is yours. Oh, and I think there's just a sound issue. You're self-muted right now, I think. Dr. Bedlack. If you can, if you can hear me, Dr. Bedlack, you just, there we go.
How about there? Are that better?
Perfect.
All right. All right.
Thank
Sorry
you so
about
much.
that. Now let me see if I can find my PowerPoint. There it is. Well, thanks so much for having me, David. I've never traveled so far south to speak to an audience that was so far north. I'm actually here in New Orleans, Louisiana, to speak to a group of patients tomorrow. But I'm really glad I could work this in. And to your question about what I'm wearing, I am actually wearing a T-shirt by a Canadian designer, pop artist named Grimes. So that's to honor you. So the title of my talk today is Lazarus by Lunasin and Untangling an ALS XFiles. So as I've said to some of you before, my work is a lot like the old XFiles TV show. And for those that don't remember this, this was a show that ran for most of the 90s. It made a little comeback last year. And it's centered on a character by the name of Fox Mulder, who, when he was a kid, had his baby sister Samantha disappear.
Rick,
and the way he would...
Sorry, I didn't mean to interrupt. Are you showing the, do you have two screens going? I think right now we're not seeing the presentation view. So I just, in case...
Let's see what I can do.
I think in the top right it offers another screen view. Is that the top right? Top left? No, that's the right.
It says here that I'm showing the screen.
Okay, it says he's showing the screen, but it's showing, it's not showing the presentation. It's showing just the actual PowerPoint. Okay, let's see what we can do here. So right now you're running the actual PowerPoint presentation?
Yes.
Oh, okay.
So you can't see my slides?
Well, we can see your slides as they would be in the presentation view, but not when you actually click on the... have you moved to slide 2 yet? I guess that would be the question. Yeah,
I'm on slide 2, the XFiles slide.
We are not
Can you see that?
No, that was why I just wasn't sure. Okay, on the panel, where would it say, Colleen? On the panel at the top? Yeah. Okay, do you have two screens going right now, perhaps, Dr. Bedlack?
Not that I can see.
Okay. Let's see. What do you think, Colleen? Okay. Okay. Yeah, you could do that. You could go slide by slide out of. Oh, we had just seen it. So maybe if you oh, there it is. We see it now. Perfect. Sorry to interrupt you. Perfect.
No
we
problem.
got it. Okay. Thank you so much. I'm muting myself. Let you get back to it.
Okay, so back to the XFiles. This show is centered around a character by the name of Fox Mulder. And when he was a kid, he had his baby sister, Samantha, disappear on him. Now, the way he remembered it, she was kidnapped by aliens. Of course, nobody believed him. And that skepticism fueled his lifelong quest to prove to the world that aliens and other supernatural creatures were real. And so every episode of the X-Files would start with some strange occurrence. Mulder and his team would investigate, and usually they'd find some rational explanation. So I had my own Samantha. This was a 60-year-old university professor who came to see me with a pretty obvious case of ALS. She was early in the course of it the first time I saw her, just a little slurred speech, easily intelligible, a little more arm and leg weakness, but still independent in her activities of daily living. Had no obvious cognitive or behavior problems. After I talked to her a little bit about this terrible disease, I was able to present her with evidence-based, stage-appropriate options, including riluzole and multidisciplinary team care. I also asked her if she wanted to be in one of the research studies that my clinic had open. Much to my surprise, she said she'd be happy to take the riluzole and come to clinic, but she was going to pass on the research study. Instead, she was going to try something that she saw on a YouTube video, which was called Lunasin. Now let me keep my fingers crossed that this video will actually play.
I think they can sort of hear it, Rick, but it's just a little bit quiet, so I'm not sure if there's a way to up the volume.
Mike had a feeding tube and couldn't lift his hand. He had his 5 children come to the house basically to celebrate Mike's life and say goodbye to him. And I never dreamed that really, at this point in time, a year later, that he would be still alive. Alive and improving every day thanks to this, a component of soybeans called Lunasin, known as a superfood, discovered years ago by Dr. Alfredo Galvez.
Prevents the normal cells from having cancer. The University of Illinois found it was anti-inflammatory. The Duke University and Indiana University, they've done experiments showing that Lunasin was improving the immune system.
Dr. Galvez says its laundry list of benefits are some of epigenetics, science's latest buzzword. Kind of like a genetic light switch for your DNA, turning unhealthy genes on or off depending on your lifestyle choices.
So by treating the epigenome, then you can reverse the chronic diseases. We're not advocating developing new drugs. We're just advocating optimal nutrition.
For Mike, that means 10 Lunasin capsules every day, along with these nutritional products added to daily shakes, none of which are regulated by the FDA.
I think we have to be careful. Is it bad for you? Probably not. But we really don't know.
For the Macduffs, Mike is all the proof they need that something as simple as Lunasin can work.
I started at swallowing again. I gained weight. I gained 20 pounds.
And he's gaining strength.
Instead of getting worse and worse, he's getting better and better.
No prescription needed for Lunasin, but it is not cheap. It costs $80 for a 120 capsule bottle.
Well, I hope you'll agree that that's a pretty interesting video. For the purposes of this talk, Lunasin is an example of something that I'm going to refer to as an alternative or off-label therapy. I'll abbreviate those AOTs and define them as treatments advertised to slow, stop, or reverse ALS without what most of us would consider good scientific evidence. It turns out that my Samantha is not the only patient who's considering something like this. Surveys suggest that at least 50% of the pals that are out there will try one or more AOTs over the course of their illness. Why? Well, the things that we have to offer them, and even the things that we have in our research pipeline, some of which we've heard about over the last couple of hours, they might slightly slow the progression of the disease and they might improve quality of life. But like the slogan on this old X-Files poster, patients and families want to believe that there's something better out there. And if you ask them why they're trying these things, 10% say they think they'll find a cure, 20% think they'll find something to make them stronger, 50% think they'll find something to slow their progression. Onto that difficult background comes the internet, where there's a massive number of AOTs for ALS being advertised. If you do a Google search of ALS treatment, you'll come up with more than 50 million hits. The other thing about the internet is that there's no such thing as internet police. So if you're selling something, if you're a proponent, somebody selling an AOT, you're allowed to make some pretty amazing claims about it. And here's some examples from a screenshot of a now defunct product that was out there called MotoTab. And you can see at the ends of the blue arrows what the proponents of MotoTab were saying. This is the world's most relied upon and clinically proven treatment for motor neuron disease. It's completely guaranteed. You're at no risk from trying this. Now, what kind of evidence is offered to support claims like these? Well, in some cases, none at all. So if you were to click on any of these links on the old MotoTab website, you would just come to a dead end. Far more commonly, though, anecdotes, short stories about people who tried the AOT and had something amazing happen are offered as evidence. So that video that I showed you, the Mike Macduff video, that's an example of an anecdote. Here's another example. This one comes off the website for a company called Genervon that has an AOT called GM604 that it's currently selling to patients with ALS. And this anecdote says, after reviewing this patient's past medical history, it's in the treating doctor's opinion that the patient's greatly benefited from GM604 over the past 6 weeks. During our initial assessment, this patient had rapid tongue fasciculation, but this decreased dramatically with the medication. The patient's also stopped biting her tongue. This is a reversal and unexpected benefit from the medication. Moreover, this patient's limb progression has completely plateaued. Well, again, that sounds interesting, but there are some potential flaws with that anecdote, right? First of all, we don't know who this person is or how their ALS diagnosis was confirmed. There's an old New Yorker cartoon that says, on the internet, nobody knows you're a dog. It turns out to be even worse than that. There's at least one example of a company having made up anecdotes to sell its product. This was the Lifestyle Lift Corporation that was sued, and when they went to court, multiple employees testified that they were told to just make up stories about fictitious people who tried the product and had some amazing benefits from it. Another problem with anecdotes like this one from Genervon is that the outcome measures that were given are often of uncertain clinical significance. So this anecdote talks about fasciculations being reduced. Well, in my experience, fasciculations are not really usually something that patients complain about that they want treatment for. Also, we know from this article here from Dr. Sorensen and Dalby that fasciculation frequency doesn't really correlate with the speed of progression, nor does it correlate with where the disease is going next. So it's not really clear why a reduction in fasciculation frequency would be clinically meaningful. The biggest problem, though, with ALS anecdotes in particular is that they fail to account for the natural history of this disease. So the Genervon anecdote talks about Plateau, a period where there was no progression and weakness that lasted 6 weeks. Well, that would be interesting if the progression of ALS was always a straight line in every patient, that every day they were a little worse than they were the day before, but it's not that way. So last year we looked in a big database called PROACT at several thousand patients who had serial ALS FRS-R scores. And a lot of them look like that one in the upper right-hand corner of this slide. So that's one patient in the database, and that's their ALS FRS-R score over 600 days of follow-up. And you can see that it's not a straight line down. There's a period between day 400 and day 550 when they were completely stable. And at the end there's even a period when they look like they got a little better for a while. The second part of this graph here in the lower right-hand corner is a plot of the percentage of patients in PROACT that had different degrees of ALS FRS-R stability lasting different amounts of time. So if you just look at the dark blue, the Duke blue trace there, which is the highest one, that's the percentage of patients who had a stable ALS FRS-R score lasting different amounts of time. And as you might expect, a lot of patients had a stable ALS FRS-R score over a very short period of time, like less than a month, and not very many had a stable ALS FRS-R score lasting more than 300 days. But relevant to this particular anecdote, we can ask ourselves what percentage of patients in PROACT had a stable ALS FRS-R score that lasted at least 6 weeks, and it turns out to be almost 80%. In other words, what that Jenner von anecdote is talking about is just probably just the natural history of ALS. It can't be used as evidence of treatment benefit. Now, if you're out there and you didn't see those flaws in the anecdotes, don't feel bad. There's a Scientific American article that says most non-scientists fail to see these flaws, and many people equate anecdotal evidence on par with the evidence that comes from randomized trials. This article even goes on to speculate a possible reason: evolution. It says we've evolved brains that pay attention to anecdotes because false positives, believing there's a connection between A and B when there's not, well, that's usually harmless. But false negatives, believing there's no connection when there actually is, that mistake might take you out of the gene pool. Now, whatever the reason, the consequences of failing to recognize these harms is pretty clear. Failing to recognize these flaws is pretty clear. Patients and families might be overestimating the potential benefits of AOTs based on anecdotal evidence. At the same time, they probably are underappreciating the harms that can come from these things. And there are real harms. There's clearly been financial harm. Some of these things cost hundreds, some cost thousands, some cost hundreds of thousands of dollars per treatment. There's clearly been physical harms. We found evidence of patients who suffered infections and blood clots from some of these things that are advertised as perfectly safe. There are examples like this article from the New England Journal of Medicine of people going and practicing something called stem cell tourism, where they go and buy stem cells from unregulated stem cell clinics, and they come back and they wind up with tumors comprised of someone else's cells. There have even been deaths in some of these clinics that were advertising themselves as safe. Finally, there can be scientific harms from the pursuit of AOTs, and we saw that with Samantha. We've got a pretty low enrollment rate in our ALS clinical trials. We average about 2 patients per site per month in these trials, and there's a lot of reasons why we don't enroll better. First of all, our trials aren't always that easy for patients to get into. They aren't always that attractive to patients. They might have lots of visits that patients don't want to put up with. They might have placebo groups that patients don't want to put up with. Patients are frustrated that it takes so long to get answers from our traditional ALS trials. But clearly one reason is that patients sometimes turn down a trial to pursue one of these AOTs. Just as an aside, I think it's interesting that folks seem to pay less attention to the evidence of harm that's out there, because that evidence is often anecdotal as well. And there's a couple of reasons why I think it happens. The first is this quote that I've heard over and over again from patients who tell me, "Doc, I already have ALS. What worse could possibly happen to me?" And as I showed you in the previous slide, there are worse things. You could have ALS plus leave your family with a big debt from pursuing some of these things. You could have ALS plus an infection or a blood clot or a tumor from someone else's cells that you didn't need to have. The other problem is that anecdotes describing harm are not as easy for patients and families to find on the internet. I think there's a reporting bias. People are much more likely to go into chat rooms and talk about perceived benefits from an AOT than they are to talk about perceived harms. These types of anecdotes are not on proponents' websites. They're scattered across many different internet locations, including different chat rooms. Some of these types of anecdotes are in the scientific literature, but that's not always easy for patients and families to access. So if a patient or family member wanted to read about what happened to some patients who went to this stem cell clinic in China a few years back, and they wanted to read this particular article that's on the right-hand side of this slide, they would actually have to pay $54 to get access to that article. So frustrated by all that, in 2009, I got together with some of my colleagues in the ALS research group, and we put together this program called ALS Untangled. And what we wanted to do as a goal was just really try to figure out a way to systematically assess these AOTs and help patients and family members make more informed decisions about them. Our motto was kind of like that of Fox Mulder and his XFiles team: the truth is out there, though we know it's not always easy to find. So there's 3 parts to this program now that we created. The first is how we get our ideas. The second is how we do our reviews, and the final is how do we get the information back to patients and families. So we wanted to make it as easy as possible for folks to get us ideas. And so these can come in through face-to-face visits with any ALS Untangled team member. They can come in through emails. All of our email addresses are out there on the internet. Most commonly, they come in now through Twitter. And so the nice thing, as many of you know about Twitter, is you don't need to remember any specific email address, just a keyword. In this case, the word is ALS Untangled. So if anybody anywhere in the world sends a tweet out that has the word ALS Untangled in it, I can find it and I can put their idea onto the open review section of our ALS Untangled website. Another nice thing about Twitter is that you can get an idea of how many people are interested in what you're doing by the number of Twitter followers that you have. And I was very excited earlier this year when we crossed the 3,000 follower mark. Went home and told my wife about it, and she reminded me that Lady Gaga has 44 million Twitter followers. So that kind of put that in perspective. But in fairness, she does have her own stylist, David, and I've got to put these outfits that I wear together myself. You might not be surprised to hear that with all these Twitter followers and all these tweets coming in, that the number of things that people have asked us about has really exploded. We now have more than 300 AOTs that we've been asked to review. We've got so many that we've had to put a vote button next to all the open ones so that we can figure out which ones people are most interested in and focus our efforts there. And as you can see, some of these things have more than 1,000 votes. Luckily, it's not just me doing this. I've got a team. The team now consists of more than 100 clinicians and scientists from across 10 countries. We've also put together a standardized way to do this, standard operating procedures that guide everything that we do, from how we gather information to how we write these things to how we actually crowdsource and ultimately publish our drafts. All of our reviews revolve around something we created called the Table of Evidence. So we are now grading each AOT across 5 categories: mechanistic plausibility, preclinical models, case reports or anecdotes, trials, and risks. And in each of these categories, we assign a letter grade ranging from A to F. A is the best, F is the worst, and there are specific cutoffs for moving from one grade to the next. And so if you look just at the case reports and think about what I was saying with regard to anecdotes, one of the things that we can help patients and families do with this method is to validate and centralize both positive and negative anecdotes and to temper their conclusions based on what we know about the natural history of ALS. So once we get a draft that we're all happy with, we send it on to the journal ALS and Frontotemporal Dementia. It gets one final review by Dr. Orla Hardiman, who's the editor, and then it gets published in the journal. All of our articles are published by free open access. No one ever has to pay to read one of these articles. And they're also published on our website. We now have 41 of these articles published. And these are not all that easy to do. Each of these articles has taken me about 40 hours of my time to complete. And so it would be fair to say that's an awful lot of effort. I mean, how do you know that this program is really worthwhile? Well, I don't know for sure because I don't know how to measure the success of the program. I can tell you a few things though. First, I can tell you that people are certainly reading these articles. So the ALS Journal keeps a running list of the most downloaded articles in its history, and the top 9 most downloaded articles in the history of our journal are all ALS Untangled reviews. And you can see that some of these articles have more than 20,000 downloads. So while I might have published more prestigious articles, I doubt that I've ever published any other article that was read that many times. Another thing I can tell you is that I've learned a lot from running this program in the past 8 years. First of all, I learned that these proponents, these people that sell these things, they're not all snake oil salesmen like I thought they might be when I started. Most of them, in fact, are what I call true believers. They really have a good heart. They think that they have something that's helping people. They just don't have any interest in or maybe any knowledge for how to prove it. Also, you know, in meeting a lot of these proponents, I can tell you that they do things that patients and families really like and that I think mainstream doctors need to learn from. They're incredibly optimistic. They're very respectful of patients' ideas, and they're very responsive. I remember spending a weekend with one of the doctors who ran a chelation clinic in South Carolina, and just being impressed that no matter how late the clinic ran, he always made sure that every telephone call and email was answered before he went home for the day. And I think that we need to really embrace these qualities for our patients. When they're dealing with a disease that's as frightening as ALS, we need to try to create a positive mood, be respectful of their ideas, and get back to them with answers to their questions in a timely manner. Another group of lessons that I learned are lessons about the treatments themselves. And again, I was skeptical when I came into this, But it turns out that many of the things that patients and families have asked us about, including some of the strangest sounding ones like fecal transplants, have plausible mechanisms and probably should be further studied. But without a doubt, the most amazing lesson that I learned is that Mike Macduff's dramatic ALS reversal that was described in that YouTube video, I think it's real. So he's got a history that's fairly typical for ALS. He's got exams that are fairly typical for the lower motor neuron form of ALS, which is called progressive muscular atrophy. He's had a reasonable workup for ALS mimics. And in looking through his medical records and talking to the doctors and the various therapists that have taken care of him, he really did get dramatically better on validated measurements of his speech, his swallowing, and his strength over the course of a year. He even improved his ALS FRSR score by 8 points over the course of 12 months. And Mike's not the only one. In the years since I've been looking for these, I found 33 other examples of what I call dramatic ALS reversals. These are people whose medical records I have, and looking at them, I feel like more likely than not, ALS was the right diagnosis. They all progressed for a while, and then they all had a dramatic improvement in at least one recognized objective ALS outcome measure. So the question comes, what do we do with these folks? Well, in my opinion, what happened to them is so different than what was supposed to happen that we have to study it and try to figure out why. Now, one possible explanation is that some of the treatments that these folks were on, like the Lunasin that Mike Macduff was taking, maybe they actually worked. And it wouldn't be the first time that patients unexpectedly and accidentally discovered a beneficial effect of a medicine or treatment that doctors and scientists had not hypothesized. It's called serendipitous drug discovery. There's a whole paper written about this. The most famous example is the example of Viagra. The company Pfizer was testing that drug to see if it would work for heart disease. It didn't. It would have been thrown in the garbage except for the fact that men in the study asked if they could stay on it at the end. And when the company realized what was happening, they shifted gears and started to study it for erectile dysfunction. It became the first FDA-approved drug for that indication. It became a billion-dollar blockbuster. In ALS, we have a similar example with the drug Nuedexta. We were testing that to see if it slowed ALS progression. It didn't, but people spontaneously came forward in the study and said, hey, I think this is helping, that uncontrollable laughing and crying, that pseudobulbar affect. The company shifted gears, tested it for that, and voila, we have the first FDA-approved drug for pseudobulbar affect. So based on this precedent, I'm actually starting a program called Replication of ALS Reversals. It's abbreviated ROAR. It's a program of small pilot trials of these AOTs that are associated with dramatic ALS reversals. Now, because most of these AOTs are regarded by regulatory agencies like the FDA as something called GRAS, generally regarded as safe, and also because we're looking for a huge signal, the largest signal ever looked for in an ALS trial, reversal, my trials can have some interesting features that I think patients and families will really appreciate. We can have very wide inclusion criteria. We don't need placebos. We can use historical controls. We don't need a lot of in-person visits. We can do virtual visits where we teach people to measure things and we teach them how to use a program, patients like me, and record the things that we want them to measure. Another advantage of doing things this way is that anybody can look in and see what's happening to the patients in our study in real time. You can go onto the Patients Like Me website and type in ALS and Lunasin, and you can see what everybody who's taking Lunasin, including the patients in my trial, are saying about it. And last but not least, we're actually going to publish our protocols. So when our protocols get IRB approved, we're going to put them on our ALS Reversals website. And this way, patients around the world who are going to self-experiment anyway can be empowered to self-experiment with something that might have a little more plausible rationale, with a specific source of the product, with specific dosages, and they can even measure things and record them on the same website, Patients Like Me. And so the first of my ROAR trials is a trial of Mike Macduff's Lunasin regimen. And before we went into this trial, we actually did an ALS Untangled review of Lunasin, and we found that it did have a plausible mechanism. According to the company, it does alter histone acetylation and subsequently alters the pattern of gene expression in cells. Histone acetylation is known to be abnormal in patients with ALS, and that is part of why we think that they might have an abnormal pattern of gene activation. And we've gone after this mechanism in a mainstream ALS trial at least once before. We did a trial of something called sodium phenylbutyrate to try and alter histone acetylation. So we gave it a mechanism grade of C. In addition to Mike Macduff, there are other people around the world, 13 out of 17 when we started this project, that said they had taken Lunasin for ALS and had improvements. We never could get their records. So we only have one validated case with a diagnosis and improvement, so it gets a case's grade of C. And at the time that we started this, it seemed that Lunasin was reasonably safe. We couldn't find any serious adverse events associated with it, and really side effects of any kind appeared to be rare across the internet, so we gave it a really good safety grade. So the ROAR Lunasin trial really had sort of two groups of hypotheses. The first group is related to the treatment itself. We wanted to see if Lunasin was safe, tolerable, could alter histone acetylation, could increase the frequency of ALS reversals, and could slow progression by at least 50% relative to historical controls. And then there were a group of hypotheses related to this unusual design. We wanted to see if this design was associated with improved enrollment rate and better retention compared to more traditional ALS trials. And we wanted to see if the folks in this trial could actually accurately measure their ALS FRSR scores and their weight, and if they would go on to patients like me every month for the course of a year and record those scores. So just a little bit about the design. It's widely inclusive, it's largely virtual, it's one site, it's a historical control group. We're enrolling 50 patients, and that's based on our power calculation. You can see the inclusion-exclusion criteria on this slide, and I hope you'll agree that these are much more broad than our typical ALS trials. And again, it's a 12-month open-label study. There's only 3 visits over the course of a year. We had to have people come in 3 times because we wanted to double-check that their measurements agreed with our coordinator's measurements, and also because we wanted to gather blood to look at histone acetylation. Most of the visits were virtual. Again, people were logging on to patients like me and entering the things that we taught them to measure: their ALS functional rating scale, their weight, their perceived effectiveness, their concomitant medicines, any adverse events that they perceived, and their compliance. And we had some coordinator calls, which were used to be sure that patients understood how to use the website and understood how to enter the data that we talked about. So the trial's still going on. We still have a few patients in it. Almost everybody's finished, but there's still a few, and the last patient's going to finish next month. But I can present to you today some interim results. We enrolled 50 participants in a little over 5 months, which, as far as I can tell, is a new enrollment record. As I mentioned, that's almost 10 patients per month. Most trials enroll about 2 patients per site per month. As of this week, we've got 26 people having completed the entire 12 months of the study. 9 are still in it. 15 have terminated early, 6 due to death and 9 due to dropout. And most year-long traditional ALS trials have a dropout rate of around 30%, so we're doing better than that. Our measures of compliance with treatment and data entry suggest that the compliance and data entry are very high, and the outcome measures have excellent agreement between what our coordinator is measuring on the phone and in clinic and what patients are entering on the Patients Like Me website. In terms of safety, we've got, again, 2 sort of ways of looking at this. One is the traditional measurement of AEs and SAEs, which occurs by our coordinator over the telephone and in clinic. And that's on the left-hand side there. That's a table of all the SAEs and AEs. And I would say that by those measures, this is reasonably safe. GI side effects are by far the most common, and in fact are much more common than we expected, and in some cases have been severe. So we've had 2 cases of really severe constipation that required hospitalization for disimpaction, and we felt that those were possibly related to this regimen. On the right-hand side there, you can see what patients are saying on Patients Like Me, and you can see that there's a pretty good agreement. So in general, most people are saying that they're not having many, if any, side effects, but there are a small number that have moderate to severe side effects. In terms of our biomarkers, we haven't been able to measure any changes in histone acetylation associated with Lunasin. Unfortunately, we've not seen any more ALS reversals like Mike Macduff's. Most of the patients on Patients Like Me are not reporting any dramatic efficacy. We'll do the analysis of ALS FRSR progression relative to virtual controls when everyone finishes the study in late September. And then there was this question that we had about playing along at home. I mentioned that we published the protocol, and we were curious to see how many people were actually trying to follow that outside the study. And there's more than 3 times the number of patients around the world who are trying to self-experiment with Lunasin outside our trial. And so we took a look at how often they're inputting data, and although they're saying about the same things in terms of efficacy and side effects, their data density is not very good. And you can ask me about this later, but this has some implications for designing large-scale expanded access programs in terms of what kind of data we might expect from those. So I haven't given up on the idea that some of the other treatments that ALS reversals have taken might work, but we do have to consider other possibilities. Maybe it's not the treatments, maybe it's something else. So a very popular explanation amongst my peers is that maybe these folks never had ALS in the first place. Maybe there's some mimicker here that was not recognized. And you know, there are potentially fluctuating antibody-mediated ALS mimics that are rarely described in literature. Atypical celiac disease, multifocal motor neuropathy with upper motor neuron signs, and even one case of myasthenia gravis with upper motor neuron signs. To me, the most interesting explanation is that maybe some of these folks have some sort of endogenous mechanism for beating ALS. Maybe they're genetically different and the ALS can't take hold in them like it does in everybody else. It might sound far-fetched, but there's a precedent for this. There's a group of people that get HIV and never get sick from it. They never develop AIDS even though they don't take any medicine. They're referred to as elite controllers. And somebody finally studied all those folks and found that a lot of them have the same genetic abnormality. It's called CCR5 delta 32. It winds up changing the conformation of a cell surface protein called CCR5 that HIV needs to get into their cells. We never knew that. We never knew that about the biology of HIV before we studied these folks. And Pfizer now used that information to develop a drug called Maraviroc, which blocks the CCR5 receptor and works for everybody with HIV. So maybe if I could find something different about the genetics of these ALS reversals, I could find a new pathway we didn't know about that might lead to a more effective treatment. And so I've got a second study going on, a second research program called STAR, Study of ALS Reversals. And here what we're trying to do is find as many of these cases as we can, get them all into the same database, and see if there's anything different about them in terms of demographics, disease characteristics, comorbidities, the treatments that they took, compared to folks with more typical ALS in large registries like PROACT and the National Registry. We're also trying to get as many blood samples on these folks as we can to look at their antibody profiles, to screen for rare ALS mimics that most of us don't typically test for, and also to look at whole genome sequencing and compare their whole genome sequencing to those of more typically progressive patients. And I can tell you this is hot off the press, which is why it doesn't look as pretty as some of the other slides. But this is the first results from the STAR study. I had a great medical student work with me over the past year, Daniel Harrison, and he was able to show that there are some differences in these 34 ALS reversals compared to patients in PROACT and patients in the National Registry. So the ALS reversals, which are referred to as cases in graphs A and B, they tend to be younger, they're more likely to be male, they're less likely to be white, they're more likely to have limb onset disease, and they're more likely to progress faster until they reach their nadir and start reversing compared to people with more typically progressive ALS. Interestingly, one thing that's not different is they have about the same percentage of family history. About 10% of the ALS reversals had a family history of ALS, just like the folks in PROACT and in the National Registry. One other thing that I find fascinating, and that is that some of the treatments that these folks were taking were used by more than one reversal, and as a result had a very high, highly statistically significant odds ratio of being associated with ALS reversal. And the one that's got the highest odds ratio was actually used by 3 of the 34 ALS reversals, and that is curcumin, also known as turmeric, the spice. And so that is now going to be the target of my next ROAR study, curcumin. So just in terms of conclusions, I hope I've shown you that when it comes to AOTs, the truth is out there, but it's not always easy to find. Anecdotal data in ALS is problematic for anyone to interpret. It's commonly misinterpreted, I think, especially by patients and families. We built this program, ALS Untangled, to try to bring patients, families, clinicians, and scientists together to systematically review and report on these AOTs. And if nothing else, we can at least help them to centralize and validate positive and negative anecdotes and temper conclusions based on our knowledge of the natural history of ALS. But it turns out that some anecdotes really are interesting and probably warrant further study, and these are the dramatic ALS reversals. And we've now got 2 research programs underway, ROAR, where we're doing pilot trials of some of the same things that these folks took during their reversal to see if any of them might work. The trial of Lunasin is nearly done. Unfortunately, we haven't seen any other reversals, but I do think that that design was a success. And STAR, which allows us to sort of explore other hypotheses related to these reversals, including that maybe they had under-recognized antibody-mediated mimics, or maybe there's something genetically different about these folks. So I'm sorry I ran a little bit over time, but I really want to thank you for letting me participate in this, and thank all my sponsors and the folks who participated in my research, and I'm happy to answer any questions.
Great, thank you so much. Really appreciate that. That's a lot of years of work, and we appreciate being able to see some of the Lunasin results as well, and what you're finding with the reversals. We do have a couple questions. The first one, actually, I'm going to throw out there relating to the curcumin. If you're going to look at that next, I'm just wondering, and maybe you don't know, or maybe you can jog my memory, if people have definitively studied, I know it's the NRF2 pathway, I think, the antioxidant response mechanism that curcumin tends to go towards, but maybe, I don't know if will there also be an untangled one for curcumin as well, or?
Yeah, so we're working on the ALS Untangled on curcumin right now, but what I can tell you is, much to my surprise, when I went to PubMed, which is part of our SOPs for doing an Untangled, is to review various databases and look for scientific publications about this thing. There's over 5,000 articles in PubMed about curcumin and turmeric, and so I've got quite a bit of reading to do to try to figure out what's the best source of these things, what's the best dose to use, what kinds of biomarkers should I be looking at in my next ROAR trial. So most of the time with Untangled articles, we have the opposite problem. There's really nothing that's been published, and in this case, we've got just a massive amount, most of it from the oncology literature, to try to sift through. One of the hard things to try to figure this out is it's amazing how little detail there is in a lot of charts in terms of the AOTs that patients take. So the 3 charts that I have on these 3 reversals that took curcumin, all it says is curcumin. It doesn't tell me exactly what brand or what dose they were taking. So I think that's something for the clinicians that are listening, is something that we all could probably do better, is to better document exactly what these folks are taking.
Sure. So in that note too, and this may be a silly question, but in terms of potential countries or ethnicities that would use cumin in a more common way in their diet, is it possible to examine there whether or not you have a higher incidence of reversals? Or maybe it's just because that's dietary versus somebody who's taking it specifically in a medicinal sense.
It's a great question. I've also been asked the question, is the risk of ALS lower in countries that use a lot of that spice? And the answer is, I don't know. I mean, I don't see a lot of ALS literature coming out of those countries. It seems to me that a lot of the countries where curcumin and turmeric are a staple of the diet, the ALS infrastructure is not as well organized as it is in other places. So it seems to be much harder to get data from there.
Of course, sure. Okay, so I have another question. Looking back at the Jenner Vaughan story and GM604, the way that advanced in terms of coming to the public and getting the public's energy behind it came in a very different way from many AOTs in terms of they ran a clinical trial with some very seasoned investigators who were doing the administration, and then did basically, as you said, an internet press release type situation. Do you foresee this potentially being a problem in the future, where people are thinking about new ways in which they can, get some legitimacy behind what they would like to sell or believe in?
I think it's a reality now. I think that we live in the age of spin, and I think that This is something that like it or not, we're all going to be asked by our patients and families to critically examine a lot more stories, including stories from manufacturers of products around the world that are out there. And I mean, I just think that's, hopefully ALS Untangled, we can continue to ramp up our efforts, because I think that's one thing that those articles can do is help a busy clinician. You know, if a patient asks about something that we've already looked at, you can just pull the article. But, you know, for all of us to take 40 hours and do an in-depth examination of every story that's out there, it's going to be difficult. But we do live in an era of spin, and I'm seeing more and more interesting things, even from, you know, companies that appear to be mainstream. The way that they're spinning their data on the internet is really confusing to patients and families.
Sure. No, and I'll be the first to admit that I'm so thankful to have ALS Untangled to point to in a lot of situations where you know, someone will ask me about a gluten-free diet or something like that, and I can, or the Deanna protocol, and I can, you know, instantly say, here's a very good document that you can access and be able to read it. One more question, and this is a kind of an interesting one. I don't know. Well, I don't know if it's that interesting. I made it up, so maybe not. But so thinking towards the future, we're getting possibly to an age where there may be some therapies that work for ALS in a modest sense that could come down the pipeline, and then hopefully, you know, eventually we get to the point where some of the advances that have happened preclinically the past 5 years start to make real therapeutic targets that have a more substantial effect. Thinking towards those therapies coming out, having modest effect, potentially being very expensive, do you see that what are your thoughts on having more traditional therapies available, possibly reducing the amount of AOT use, or is there the possibility that if people are having to pay out of pocket for for very expensive therapies, that suddenly some of these $30,000 stem cell tourism things become the cheap option, and people continue to think that it's an option to go towards?
No, it's a terrific question. I mean, my opinion is that the frequency of self-experimentation with AOTs is only going to increase over time until we find something that, you know, stops or reverses ALS. So, you know, each time a new treatment comes out and gets FDA approved, you know, when people look at the reality of the cost and the difficulty of it, there are always spin-offs that claim, well, this is cheaper and easier, and it's just like, you know, product X that was just FDA approved, so why don't you use this instead? And so I think in a way it almost is going to make the problem even bigger. I think the success of treatments that are modestly effective but difficult to use and expensive could make this even a bigger issue over time.
Actually, that's a really good point too about the mimic thing. I'm wondering if, you know, now that there's another drug approved in the United States for ALS, if suddenly, you know, people start to look at what's the proposed mechanism for this and saying, well, we've got something that does that same mechanism and it's way cheaper, and so I could see that actually opens up a whole new avenue of needing for diligence on the behalf of the field to sort of manage.
Exactly.
Well, thank you so much. We really appreciate this. This has been great, and I hope you have a wonderful rest of your day in New Orleans and tomorrow with the people you're speaking with, and we really appreciate your time.
Thank you, David.
All right, take care. Thanks.